I'm going to use the very first slides. As you all know, that I just in-house investigators. Our lab just moved from Northwestern to UAB. Now we have been building up the team. Now have this many investigators and 3 incoming. Have research faculty and postdoc and PhD student, and also research technician level, different level of investigators. So we have now a very powerful, strong investigative team. So this we're calling for investigations. So firstly, just a very brief overview of what we are doing. And basically, we are working in the ischemic heart disease. Our lab starts off studying the coronary microcirculation and cardiac protection and angiogenesis. I think I can probably, yeah, a little bit. Okay, and then the genesis was better, right? So then we started studying the stem cell biology, focus on blood circulating endothelial progenitor cells, different sorts of bone marrow stem cells, and moving on to years iPS cell biology, and also we culture certain stem cells from the adult heart and the so-called cardiac stem cells, and study the physiological relevance and the therapeutic implications in the small animals model and the translational preclinical animal models. And because basically we are more focused on the molecular and the cellular mechanism of biology in this disease setting. So for our very molecules we are very interested, we actually go further, study their implications and their biological functions in the vascular system, such as in the vascular inflammation and hypertensive disease, and even go further that in the we find some different phenotype. We go further in the adipose thermogenesis, obesity, etc. So basically, these are either the cause of ischemic heart disease or basically the major risk factors. So this is our area of research. So today I'm going to focus a little more about stem cells. Okay, this particular aspect. So I mean, before I go into that, I think I've been frequently asked, whether you think that cardiovascular regeneration is a matter of cardiomyocyte renewal, proliferation, or is a part of the real stem cell mediated regeneration. So my answer would be that maybe both actually have good evidence, including the endogenous stem cells, maybe involving the cardiac renewal. But not today. I'm just focusing on this particular aspect of cell therapy. So for cell therapy to be successful, we have to overcome certain challenges. For example, whether the cells can be delivered right place, right time. And if we use endogenous stem cells, whether stem cells can be mobilized selectively into the circulation or be recruited selectively to the ischemic injury. And so in our previous study, we have been focusing a lot how to selectively mobilize bone marrow-derived endothelial progenitor cells. So for us to do that, we have to understand how stem cells reside in the bone marrow microenvironment. So we have done a lot of work in this aspect, focusing on this the molecular interactions between stem cells and this microenvironment. And especially we established that the crosstalk between this integrin signaling, the chemoattractant signaling, and the receptor tyrosine kinase, which how they actually crosstalk in this very unique specific context, which they may not necessarily in a different context. And we also focus pretty much a lot on once the cells are recruited in this chemical injury, how they do to regenerate. So what's the signaling to make them, to attract them to the place, and how they migrate, how they interact, and how they invade the tissue that can make them go further beyond the place that the vasculature can reach. Because angiogenesis can happen, and then they can, starting from the branching of the existing vessels. So for the stem cells, we believe vascular stem cells can do something more differently. So we have done a lot in this aspect. Still until a few years ago, I didn't have the very recent data showing that even now, the majority of the clinical trials are still using bone marrow derived cells, that the majority. So these cells that's in a very recent publication, I guess this group has a new updated version, but the message is the same. Basically, they compile, about a lot of, almost all of the eligible clinical trials and analyze what's the effect. They find that actually bone marrow derived cells in all these blinded, well-controlled trials, they find that cells really can improve LV injection fraction, reduce infarct size, and ameliorate adverse remodeling. So, it's fair to say that the overall is relatively positive, it's safe, but it's not, of course, not terribly effective. So, because of these challenges. So, in addition, since these cells have this capacity to be able to differentiate, so I think today I'm going to focus on addressing these challenges. What's the plasticity and the stem cell dysfunction? The reason I'm doing that is because, for this very earlier report from Science from my colleague that published about 20 years ago, showing that this bone marrow derived population of cells that when culture and the proper condition, they can differentiate into endothelial cells and become part of a new vasculature when angiogenesis is required. So basically, they show that this is the cells have the capacity to differentiate to vascular cells. So why this is becoming important? Because recently, if you after so many trials, if you ask a clinician why the cell therapy does not work, they say, no, they do not stay for a very long time. But what does it mean? It means that they may not be physically become vasculature, incorporated into vasculature. So we think that for them, for this therapy to be working, that in addition to a lot of function, we are assigned to these cells like paracrine, like exosomes, a lot of other activities that I think is kind of fundamental for these cells to be able to differentiate into the cardiovascular cells. And so what prevents them to do that? So I think recently we made some finding in this aspect because of our study on UTF1. This study can shed some light how in the adult tissue we can make these cells more prone to differentiating to cardiovascular cells in addition to their paracrine effect. So this UTF family of transcription factor our lab has been also working on in the context of coronary microcirculation angiogenesis. And we find that UTF1, 2, 3, as you all know, are active cell cycle regulator, and 4 to 8 are negative regulator. Basically, this G1S phase checkpoint control of cell cycle, right? It's very important. But we have been focused on this family extensively for the last over 10 years. We find that actually, even though UTF1 is a positive regulator of cell cycle, but with the knockout, endothelial specific knockout, in the inhibition, we find actually basically knockout UTF1, it doesn't have any effect to affect cell cycle in the endothelial cells. Instead, actually it has a enhanced angiogenic phenotype. And the ETOF2 knockout, you don't see any problem in angiogenesis and endothelial cell growth. Instead, we find that these mice display increased blood pressure. And only ETOF3 knockout seems to be have very severe cell cycle problem in the vasculature. They have a significantly reduced angiogenesis and neovascularization because of the endothelial cell cycle. So come back to this ETOF1. So we have very firstly reported that knockout ETOF1, they have an increased angiogenesis. So this is a hand ischemia model. We resect the femoral artery, look how this blood vessel grow back, find that this knockout mice blood vessel growing back is much faster and then the wild type. So this is interesting, and actually this report has been confirmed independently by different group from University of Washington, and also in a different model. This is the ischemic reperfusion model. As you can tell in the ETOF knockout mice that they have a much smaller infarct size than the wild type, and also we perform a total ligation model and myocardial find the same thing. So basically, ETOF knockout is protective of this ischemic tissue injury. So because we're interested in stem cells, so she asked whether ETOF1 has a role in the bone marrow stem cells. So basically that he crossed the ETOF knockout and the GLP transgenic, have this two duogenic mice, and perform the bone marrow transplantation with this bone marrow into wild type and control into wild type. Compare after recovery of bone marrow, then induce the myocardial function in the recipient, then look at ischemic heart, how many bone marrow derived cells show green and how many cells become vessel, which is red. So he found that in the mice that received this ETOF1 knockout bone marrow display much more that vasculature derived from bone marrow cells. So this is very significant. So then he went further to analyze the gene expression of these bone marrow stem cells. And he did at that time he did a microanalysis. Out of actually many genes changed, that about 75% of the genes are actually in the category of metabolic genes. So this is very surprising. There is no change in any cell cycle genes. So this is very surprising. That it seems that ETOF1, the major role in the stem cells is metabolism. So basically, we took some time to dissect out what's the problem. Finally, I don't think we did, but we did find that this PDK2 and PDK4 is a pyruvate dehydrogenase kinase is significantly reduced PDK2 and PDK4. So confirmed by RT-PCR and by Western blotting. So since that there's a PDK2 and PDK4 is inhibitor of pyruvate dehydrogenase. So basically we have a reduced PDK2 and 4. We would expect to be a less repression on the PDH. We would have an increased production of acetyl-CoA. We would expect to have an increased mitochondrial oxidative phosphorylation. This is actually exactly the case. And then we isolate the bone marrow stem cells, analyzed by seahorse the oxidative metabolism. We find that in these stem cells, they have an increased oxygen consumption rate, very significantly, and also have a reduced extracellular acidification rate, indicating that anaerobic metabolism is actually reduced. So this is exciting, we think that, because we already know that stem cells primarily rely on the glycolysis. A lot of times it's anaerobic glycolysis. But with the differentiation, when they become function mature, they actually we thought they adapt to a state of which more relying on the mitochondrial function, oxidative phosphorylation. So now this model provides us with very good model to study whether actually metabolic change, the shift, the switch itself actually is a driver to make the cells more prone to differentiation. So this is very interesting. So we then went further to test this hypothesis. So we put these cells in the culture that knockout the stem cells, white type stem cells, in the endothelial special culture. We find that after 7 days, these knockout cells are much easier to become endothelial cells to differentiate. And also we overexpress that this might not be the best way, but because if we're not downregulating our PTK4 is the reason now we tried in the knockout cells to overexpress PTK4, actually this can reverse the metabolic alterations, basically. And it looks like, it might not be the best experiment, but this seems to be E12-1 mediated metabolic regulation seems to be because of the reduction of PTK4 and PTK2. So basically now for the differentiation in the knockout, and so we have an increased differentiation as indicated by CD31 staining, but if we overexpression PTK2 and PTK4, this increase become not significant. But if we actually reduce this one, also not significant. So seems to suggest that the PTK2, PTK4, and the subsequent PTH changes is the reason for the differentiation alterations. So we even went further and identify the E12-1 binding site in the PTK4 promoter. We actually by mass spec and the co-IP mass spec, we identify a novel cofactor of E12-1. Actually, we co-express E12-1 and then this cofactor with dramatic increase in induction of PTK4 promoter activity. So it looks like it seems to be a very direct regulator, but we're still in the process of validating. And then finally, for this particular project, and then we operate on many mice to make sure. So we transplant the mice with E12 knockouts, bone marrow cells, and white type bone marrow cells. Then create the myocardial function in the recipient. We did find that if the bone marrow are replaced completely by the knockout bone marrow, we did see a significant function again after the myocardial function, here as this contractile function, and have reduced the infarct size. And also we again see that the E12 knockout bone marrow become vasculature in the heart much more frequent, and also there is an overall vascular density in the infarct and the infarct border zone. So why this is great. It looks like this is biology. This is very important. And we do believe that if you really change the metabolic states of the total bone marrow, we do see some function again, which is encouraging. So we went further, find that it's probably even more than what we thought, because because we know that in a cell therapy trial that if the patient, they are aged, they have diabetes, if you analyze their data, they are performed the worst. So as we all know that the PTK2, PTK4 is a very important target of insulin. So insulin signaling leads to phosphorylation of PTK2 and 4. Inhibits its activity. So basically, this is how insulin actually, by inhibiting PTK2, PTK4, release PTH actually increase the mitochondrial efficiency. But during the insulin resistance, so PTK2 and the PT4 cannot be phosphorylated, cannot be inhibited by the insulin. So this is a part of, so the mitochondrial function is actually reduced. Now our study suggesting that E12-1 actually transcriptionally regulates PTK2, PTK4. If this pathway operates also in the diabetes, and plus this post-translational modulation, seems that maybe the double hit to these diabetic stem cells. So we actually have some preliminary evidence to suggest that this is the case. That we from diabetic mice, we give the mice a high-fat diet. 1 month before harvesting of the sample, we actually give 1 streptozotocin, basically try to make a type 2 diabetic model in about 3 months of time. So we find that, and in the diabetic condition, we find the eTOF1 expression is significantly increased. So are the PDK2 and the PDK4. Then in the culture condition, we also put the stem cells from knockout wild type in a high glucose, high lipid. Then we mimic this kind of diabetic condition. We find that again in the wild type cells increase PDK2 and the PDK4, and actually this increase is attenuated if eTOF1 is not there. So suggesting that the eTOF1 seems to be important for the PDK2, PDK4 upregulation in a diabetic condition. So also finally we find that we have some stem cell left over from the clinical trials. We know they are diabetic patient, and then they are CD34 positive cells. We now try the knockdown of this eTOF1 in these cells. We find that after knockdown of eTOF1, PDK2 and the PDK4 are indeed reduced. So it seems to suggest in this pathway operating in the diabetic condition. So for this part of the talk, I think I introduced to you that number one, the need of eTOF1 gene is protective of ischemic and ischemic reperfusion injury, and the inhibition of eTOF1 in the bone marrow stem cells decreased PDK2, which increased the mitochondrial oxidative phosphorylation, appears to be important for endocytic differentiation, and also the deleting of eTOF1 in the progenitor cells enhances these cells differentiation and also vascular repair in the ischemic heart. And also this pathway seems to be operating in diabetic culture, diabetic mice, and the diabetic patient stem cells. So I think that for this part, I think the take-home message is that we come to learn that we have done so much how to get the cells there, and how to to mobilize them, get them there, and a lot of times they can be there, but they may not be prepared metabolically to differentiate to do their job. They are still the inertia for them to be still wants to maintain the condition in the bone marrow. So I think that maybe metabolic preparation of stem cells before we give to patients, this might be a way to actually make them more effective. So in this context, we are doing a bioengineering approach because there are PDK2, PDK inhibitors on the manufactured by the company, developed, for the purpose of diabetic cardiomyopathy, but because, for some reason haven't been gone to the clinic. So now we are trying to use this method to specifically target stem cells to on the diabetic, stem cells and the normal stem cells to improve the potency of differentiation for translational study. So the second story I'm going to tell is about stem cell survival. Okay, so this has something to do with a non-coding RNA. It's called MyArt, and so I don't think I need to tell too much about this background. We all know that in the human genome, the majority of the transcript do not code for protein products. Only a very small portion actually have protein product. Okay, so in this non-coding RNAs, some are small, we call like a microRNA, for example, and some are non-coding, more than 200 nucleotides. So, And this is a huge black box because previously, because they express in very low abundance, we thought they are just transcription noise, in the sequencing, but now obviously they still have some functions. And we don't know that much. We name them depends on where they are transcribed, whether intergenic between the genes, or introgenic, or from the enhancer. So I think that the message is that we don't really know how to characterize them. And there are some structure bases, but not well characterized. The RNA structure studies are very complicated, very difficult. This also can be spliced, act on a domain structure, and also they can mediate protein DNA interaction, protein RNA interaction. They can mediate, they have their own structures, and also they can change the conformation to interact with different partners. So, they can do many things. Basically, I can imagine they can do almost every biological, actions in a single cell. So currently, this has become also very popular in the cardiovascular field because people have continuously identified some of the non-coding RNA play a very important role in the cardiac development and then even in the disease condition. And so today, the myopathy is intergenic. And so about the whole stem cell non-coding RNA project in the lab. So basically, we do two approaches. One is a high throughput approach. Sometimes we sequence the ear cells and make them differentiate in the cardiomyocyte. So this is my cardiac progenitor cells. Now we have very well differentiation program established in the lab. We sequence the transcriptome at a different time point, and then analyze how they change according to the course of differentiation at a different differentiation stage. And also, we compare this, for example, in this case, the mouse ear cells differentiated for 8 days become a cardiac progenitor cells. We very well characterized, and then we compare this with endogenous cardiac stem cells. We isolate directly from mouse mouse heart. We analyze their transcription differences. We find, especially focus on annotated, what are the common genes upregulated as versus the primitive states, or what's the downregulated, what's commonality, and then we generate some hits that we are still following. And another approach is we use a candidate approach, basically in that we search a lot of literature, try to find what genes are cardiovascularly important, what are genes that are evolutionarily conserved, so we can predict whether they have some functionality in the cardiovascular system. For example, this myopathy, by screening we find this myopathy seems to be important in the stem cell biology. And this myopathy is called a myocardial infarction associated transcript, and so very, very well conserved in the intergenic. I feel keep very far away from the neighboring genes, and also have some role in the retinal cell differentiation. And also, very importantly, why we focus on this particular non-coding RNA is because this is the first non-coding RNA found to be related to human cardiovascular disease by GWAS, and they find that this very big study, 3,000 patients, SNP analysis, they find 6 single nucleotide polymorphism in this gene have strongly associated with the risk of myocardial infarction in humans. So this seems to be very interesting as a starting point. So for us to study that, we initially actually analyze, as everybody do, analyze expression pattern. We find that this myopathy gene is most abundantly expressed in the brain, and secondly in the lung, a little bit in the heart, but surely expressed. And the heart, neonatal heart expression a little bit higher. So basically that's the expression pattern. And this postdoc and very talented postdoc and technician in the lab have performed a lot of in situ hybridization analysis because the RNA basically exhausts different level of cardiac sections. We find that actually only about less than 1.5% of the cardiac cells actually express this myart. Now, and most of the cells are no expressor. By our in situ hybridization, express a little bit. I mean, the high pressure is very rare, so very rarely expressed. But after you induce a myocardial function, I mean, now you can see the cells expressing this gene is dramatically increased in the epicardium area, and the whole epicardium, not only the place of myocardial function. And the other place doesn't seem to change much. I mean, so this is about 15 times increase in the expression of these positive cells in the epicardium. And also I have to tell that when we stain this gene did double or triple in situ hybridization, we find that this gene expression is not limited to any cell type. It expresses these collagen producing cells, maybe fibroblast, and smooth muscle actin expressing cells, and then this DDR2 maybe or another, and also WT1 cells. So I mean, normally non-coding RNA sometimes have a lot very good specificity, but in this case, myart seems to be rather broadly expressed by different cell type, even though they are at very low abundance. And then you isolate different cell type from the heart, analyze different individual cells. So this has become very interesting because it looks like that the highest expressor of this myart is these cardiac stem cells we have defined previously. And these two types of cardiac stem cells. Others are lower. Okay, so this is the cells I was talking about. This is that we with this experiment through cardiosphere selection. We published quite a few papers on this one. So these cells express the highest level of myart. So this is interesting. Since that myocardial function is kind of inflammation, so we try to do an in vitro culture system mimic the conditions in the myocardial function. So we find that the TNF treatment doesn't change expression, but the serum starvation re-stimulation increase, and the high oxygen culture increase, and also H2O2 treatment increase. So it looks like that this myart expression in these stem cells are particularly induced in the oxidative condition. So this is very interesting. Then we, in that time, we do not have the knockout, so we are lucky to get a 2 clone, very good antivirus after selection. We get a very good knockdown of this gene, which is very lucky. And then we find that after knockdown of this gene in cardiac stem cells, we find that these cells become much more vulnerable to the treatment of H2O2. And this is just 4 hours, so it's kind of more like a cell death. So these 2 clones, it's very obvious. Oh, and also they have a delayed phase of, tunnel staining positivity, increased apoptosis compared to the non-control transduced. And also they have the increased senescence compared to the control transduced cells. So, and also we try to, put this myart knockdown cells and the non-knockdown cells, both are, labeled by GFP, into the ischemic reperfusion heart, finding that then stand for apoptosis, kind of try to mimic kind of, in vivo endogenous condition. We still find in this ischemic reperfusion heart, we put these knockdown cells are more prone to apoptosis. So basically, it seems that it's very important for the cell survival in this condition. So as you all know that cell death in in the oxidative stress, one of the hallmark is DNA damage response response. So we are not very familiar with the context. So basically, we just try go by this protocol. Company brochure looks like what's the changes in this ATM, ATR kinase, and the upstream, downstream. We try to analyze this DNA damage response pathway signature proteins to find what would be the pathway. So find that in the knockout down of these myods, expose the cells to H2O2, we find that CHEK2, this checkpoint kinase phosphorylation activation is actually come earlier and also come stronger. So this seems to be very, so this CHEK2 kinase activation is exaggerated. Then we actually performed whether other molecular, especially protein player also collaborate with myod. So we performed RNA immunoprecipitation and put on RNA, perform mass spec, we identify FAS. This particular protein seems to be a myod interacting protein, especially under the treatment of H2O2. This is a very tough experiment to do because RNA seems to be very sticky. We perform a lot of stringent watch, try to get rid of the non-specificity. So for this one, FAS is very interesting. Number one is being sure it's RNA or DNA binding protein. Number two is highly expressed in neuron. Number three also is actually oncogene. Fused with other gene can cause two types of cancers. And also there's one paper showing that seems that this molecule is a target of ATM kinase. It's a biochemical paper. So this seems to fit our overall framework. This might be our interacting partner. So basically, we went on and identified them by PCR mapping. We find that in this myod exon 7 mediated that the FAS is this place mediated the interaction between the FAS and the myod. And in the FAS, I think in this domain mediated its interaction with the myod. So importantly in that we knock down FAS, we kind of also, the cells become more vulnerable to H2O2 treatment. They're easy to die also. And also turn out same thing. They are more easy to go apoptosis. And also when we do the DNA damage response pathway protein analysis, we find that in addition to CHEK2 exaggeration of activation, also seems CHEK1 is also seems to be overly activated. And the rest of them we did not see much changes. And especially looks like, so they have a different response to the DNA damage. So actually, functionally, it seems to make sense because if we overexpress myod, have a little bit of protection, but in the knockdown of FAS, this protection is gone, suggests that it seems that myod seems to need FAS to be protective of cells. And also signaling-wise, it also seems at least in CHEK2 overexpression, we find that in the knockdown of myod increased CHEK2 phosphorylation, but it doesn't if we overexpress myod, but if there is no FAS there, it come back again. So it looks like CHEK phosphorylation, over activation seems to also depend on the FAS. So basically, this data give us that some idea to come about a new hypothesis how myod would collaborate with the FAS to regulate DNA damage response, so to protect the cells. So we think that under DNA damage, this as I show you, seems that ATM is a target, and also looks like that ATM induced gamma H2AX phosphorylation, and the PV3 seems to be dependent on FAS, and the check-to activation also is inhibited by the FAS and also by the MAIAT. So it seems that only check phosphorylation by ATM seems to be dependent on the both. So no hypothesis would be that FAS and the MAIAT interaction may form a scaffold of check-2, maybe protect check-2 overactivation through unknown mechanisms that may actually protect the cells from apoptosis. So we think this might be applied to other neurological conditions because both molecules are expressed highly in the neuronal tissue. And we know that this is important pathway for cancer. And also we think that amyotrophic lateral sclerosis, right, is a kind of oxidative stress induced neuronal surgery problem. And both of them seems to be expressed in the neuron. So we think that also oxidative stress-based disease. So we thought in this pathway may also have some indications in neuronal tissue damage. And at the end of the day, we think that this is very important because looks like these two molecules that seems to be at a hub, a very important place that to make a decision when the DNA damage happens, whether they should go into apoptosis or they should go to the DNA repair. So I think this interaction may turn out to be very important regulator in this context. In the stem cells, we want them to be there to protect stem cell, at least to survive the ischemic reperfusion, that oxidative stress injury. So for this part, I think I introduced to you that MAIAT expressed in the neonate and the adult mouse heart and is upregulated in response to myocardial infarction, primarily in the epicardium. And MAIAT is preferentially expressed in the cardiac progenitor cells and promote cardiac progenitor cells survival under the oxidative stress. And the protective role of MAIAT depends on its interaction with FAS. And this interaction will suppress check-to phosphorylation to contribute to cell protection. So this is my conclusion. And we have a very interesting things going on, exciting. We actually recently, we were, by collaboration, many things are still going on. We answer the question because it's very preliminary. And we have recently obtained this knockout mice of MAIAT. And preliminary screening of necropsy, we did not find some developmental problem in the heart, but we still work with the models, myocardial infarction, heart failure, hypertrophy model, stressed heart, to see what's its role in this heart. But it's, even though we think this gene only expressed on a population of cells, a small portion, increased by stress, but we still think that we may not be able to detect, we may be able to detect only the RNAs when they are clustered by this in situ hybridization technology. There are still possibility other cells that express very, very low level of these molecules in a soluble way, single cells. So in that way, we may not be able to detect by our in situ hybridization. So basically, we are open to the possibility of these molecules in different type of cells, even the whole heart. So we're still working on that. So finally, I'm going to now just acknowledge the people in the lab. I've already introduced them, and our collaborators and the department and the Northwestern, and for funding support. Thank you very much. I'd be happy to take your questions.